Hello, good morning all. So this is my first interaction class with you all on an online audio class. Hope you guys are sitting at a quiet corner of your house with a notebook in hand in which you all would take down the lecture notes. Our today's topic is pancreas, and with the first slide which I would focus, I would be giving you all a very detailed overall view of endocrine part of the pancreas, which is a concern when we discuss pancreas in endocrinology. We have had a different approach to pancreas when I took pancreas for you all in GIT. So I would want you all to take down the lecture notes, especially the initial overview I am going to give you all with this one slide in focus. So to begin the class, I hope you all are remembering that pancreas plays a very crucial role in controlling digestion, storage and utilization of nutrients in the body. And basically all these processes, that means the process of digestion, storage and utilization in the body, requires very high degree of precision and regulation. As you all know, pancreas has got two parts, an exocrine part and an endocrine part. 80% of the exocrine part, which we have already discussed in GIT in detail, if you all can remember that 80% of the exocrine part controlled the digestion in your body. 20% the endocrine part of the pancreas controls the storage and utilization of nutrients. So when you sum it up, digestion, storage and utilization of food is a combined effort of exocrine and the endocrine part of the pancreas. Even though the endocrine part of the pancreas is only 20% compared to the 80% exocrine part, please remember the endocrine pancreas is a very important endocrine organ as it secretes insulin. And insulin is the only anti-diabetogenic and anti-ketogenic hormone in our body. So pancreas basically secretes other three hormones also other than insulin: glucagon, somatostatin and pancreatic polypeptide. I repeat the hormones. So other than insulin, pancreas also secretes glucagon, somatostatin and pancreatic polypeptide. And if you look at the actions of glucagons metabolic actions are exactly opposite to that of insulin. We'll be dealing in detail about glucagon, somatostatin and pancreatic polypeptide in the coming up classes. So we'll go in detail to the endocrine part of the pancreas. Now if you look at the picture, in the picture you can see the gross anatomy of the pancreas in this slide. I would want you all to look at the picture, the histology of the islets of Langerhans there, and try to get what I am trying to convey. The major endocrine pancreatic hormones, which I have already told you all, is insulin and glucagon. Even though somatostatin and pancreatic polypeptide is also secreted, we consider insulin and glucagon as a major endocrine pancreatic hormones. And if you look at their actions are very rapid and they are very powerful regulators of our body's metabolism. Together, they coordinate the disposition of nutrient input from the food we take, as well as the flow of endogenous substrate by their actions on the liver, adipose tissue and muscle mass. I repeat it, insulin and glucagon, which are the major endocrine pancreatic hormones, they regulate the body's metabolism. Together, hand in hand, they make sure that nutrients are equally distributed to every cells and tissues of your body by acting on the liver, adipose tissue and muscle mass. Now if you look at the picture, you can see the strategic location of the islets of Langerhans. See, islets of Langerhans constitutes the endocrine part of the pancreas, which is secreting the major hormones insulin and glucagon. So in the picture, have a look at, that particular strategic position or location of the islets, which is actually reflecting their functional role, which we are concerned here. Now the location of the islets is in such a way that it dictates secretion of their hormones first into the pancreatic veins and then into the portal vein. That's a very important point to be noted here. So please take down the location of the islets of Langerhans. You can see that in the histology part of the islets, which has been enlarged in the figure. So the location of the islets dictates the secretion of insulin and glucagon into the pancreatic veins and then into the portal veins. Now what happens because of this arrangement or because of the strategic location of the islets of Langerhans is, this particular arrangement permits the liver to be exposed to very high concentrations of insulin and glucagon than the peripheral tissues. And you all should remember that liver is the central organ in the nutrient traffic. So I repeat it, because of this strategic location of the islets of Langerhans, the hormones secreted by the islets of Langerhans, insulin and glucagon, is directly secreted into the portal veins, the pancreatic veins first, and then into the portal vein. And because of this, liver gets the highest concentration, or the liver is exposed to the highest concentration of insulin and glucagon. And so what happens is, liver regulates its release or availability to other peripheral tissues. So basically the liver modulates the availability of insulin and glucagon to peripheral tissues by extracting variable amounts of these hormones during their first passage through the organ. Okay, now we go to the next slide. Here you can see that pancreas, now we are going in detail into the endocrine part of the pancreas. So if you look at the endocrine part of the pancreas, we just see, I've already given you all an introduction into it, but let's just reinforce the concept. Let's go through the slides once again. So you can see that the exocrine part of the pancreas, which was 80%, contained the acini cells, and the endocrine part of the pancreas, which was 20%, which composed of the islets of Langerhans, consists of 3 types of cells. The major content, or the 80% of the islets of Langerhans, contains the beta cells, which secretes insulin. Then comes 25 to 30% of the cells, which are alpha cells, which secretes glucagon, and around 1 to 8% of the cells, the gamma cells, which secretes somatostatin. So this gives you a kind of reinforcing into the top, the slides which we have already done. So now let's go to slide 3. We'll have a closer look at the structure of insulin. So basically insulin, see, we wouldn't go much into the detail of the structure of insulin, but you need to have a minimum understanding of the structure. Only if you have a minimum understanding of the structure of insulin, you will be able to understand and study the concept of mechanism of action of insulin on the cell. So for that, please keep in mind, insulin is a very small protein, or you can call it as a polypeptide hormone. And if you look at the structure, insulin, which is a polypeptide hormone, it's composed of 2 amino acid chains. A chain A, or you call it as an alpha chain, chain B, or a beta chain, which are linked together by 2 disulfide linkage. and this polypeptide hormone insulin becomes active when this linkage is strong. So when this linkage is split, insulin becomes inactive. So 2 amino acid chains, 1 alpha, 1 beta, which are held together tightly by 2 disulfide linkage, is the state when insulin is active. Moment the disulfide bridge is broken, insulin becomes inactive. Now let's go to the next slide where it talks about the release of insulin. Now the two main stimulus for the pancreas to release insulin is blood and also the extracellular fluid glucose concentration. Unless glucose is metabolized within the cells, insulin secretion cannot be stimulated. So I repeat this points once again. The two main stimulus for pancreas to secrete insulin is blood, that means the glucose content in the blood, and also the extracellular fluid glucose concentration. And unless glucose is metabolized within the cell, insulin secretion cannot be stimulated from the pancreas. Okay, now we'll move to slide number 5. And this figure, you need to look at the figure very carefully, because this is a very often asked 5 marker for you all. The figure basically explains the mechanism of insulin secretion from the beta cells in response to glucose. So please note down, the figure is explaining you all the mechanism of insulin secretion from the beta cells which is happening in response to glucose. So now have a look at the figure. You can see on the membrane the GLUT2 receptors. Now through the GLUT2 receptors, glucose enters the cell. Moment glucose enters the cell, glucose is immediately converted to glucose 6-phosphate by the enzyme glucokinase, which undergo metabolism to generate ATP. You can see that in the figure. It's given very briefly there. So what happens there is, through the GLUT2 receptor on the membrane, glucose is making an entry into the cell. Moment glucose enters the cell, it is converted into glucose 6-phosphate by the enzyme glucokinase, which undergoes metabolism to release ATP. Now what this ATP does is, this ATP inhibits K plus influx. Can you see a channel there on the membrane? So this ATP released is inhibiting K plus efflux. Now what happens when ATP inhibits K plus efflux is, it causes the depolarization of the cell. When K plus efflux is inhibited by ATP, cell is depolarized. Now what happens, the third step is, when the cell is depolarized, the voltage gated calcium channels open. You can see that in the picture. You can see the calcium is entering the cell, but the calcium is able to enter the cell only when the ATP formed inhibits K plus influx, the membrane gets depolarized, only then calcium entry can happen into the cell. When calcium is making an entry into the cell, the intracellular calcium concentration increases. Now what happens when the intracellular calcium concentration increases? This induces calcium mediated exocytosis of insulin granules. Let me make it clear again. Let me repeat it. When the cell gets depolarized, when the potassium efflux is prevented, calcium channels on the membrane open, there is a massive influx of calcium into the cell, which increases intracellular calcium concentration, and this will induce calcium mediated exocytosis of insulin granules, which leads to the secretion of insulin into the bloodstream. A similar condition we had come across at the neuromuscular junction. You remember when the calcium, same, the acetylcholine release was a calcium induced exocytosis of the acetylcholine granules, where the neurotransmitter acetylcholine was released into the synaptic terminal. Similar situation arises here. So I think it's clear. I'll repeat the mechanism again. So have a look at the figure on the membrane. So this figure is actually telling you all or it explains you all the mechanism of insulin secretion from beta cells in response to glucose. You can see the GLUT2 receptors on the cell membrane. Glucose enters the cell through the GLUT2 receptor, but immediately this glucose is converted to glucose 6-phosphate by the enzyme glucokinase, which undergo metabolism to generate ATP. Now this ATP inhibits potassium efflux, which causes depolarization of the membrane. Now when the cell is depolarized, the voltage gated calcium channels on the membrane open, calcium influx increases, which increases the intracellular calcium concentration. This increased intracellular calcium concentration induces calcium mediated exocytosis of insulin granules, which finally leads to the secretion of insulin. Hope the mechanism of insulin secretion is clear, because that is a very often as 5 marker for you all, and even if the blood glucose level regulation is asked as a 10 marker, you need to compulsory include the mechanism of insulin secretion with this particular diagram. So I'll move to slide number 6. Now the slide number 6 is also a very important concept to be understood and applied. The slide heading tells that activation of target cell or the mechanism of action. So now with the previous slide, where were we? We were, we had discussed about how insulin is secreted. Now with all that mechanism you all have understood, and you all know that the granules containing insulin by calcium induced exocytosis have been secreted, and now insulin is ready for its action in the bloodstream. Now we need to know what is this, or how is this insulin going to make the cells or the tissue to do what it wants. So how it happens is, insulin as you know is a protein hormone. It's a polypeptide hormone. And it, listen very carefully here, and it acts on the cell through a membrane bound specific receptor. I repeat, insulin, we know it is a protein hormone, a small protein hormone or a polypeptide hormone. And how does it act on the cell? It cannot directly enter the cell. So it is going to make a tissue do what it wants through a membrane bound specific receptor. So basically it is this activated receptor, not exactly insulin, which is causing the cellular response to happen. But keep in mind, the particular receptor on the cell membrane can be activated only by insulin to bring about the cellular response. Is that clear? So insulin is exerting or its actions on the tissues not by making a direct entry into the cell, but all the cells, not exactly all the cells, a majority of the cells and tissues would have a particular receptor. Insulin activates this receptor, and only insulin can activate this receptor, which will bring about the action which insulin intends the cell to do. Now you need to know a little about this insulin receptor. Now this insulin receptor which is present on majority of the cells and tissues in the body is that particular receptor gene is present on chromosome 19. So the insulin receptor gene is present on chromosome 19. An insulin receptor is basically a combination of 4 subunits, 2 alpha subunits and 2 beta subunits, which are held by disulfide bond. So I repeat it, insulin receptor gene is present on chromosome 19, and the insulin receptor has got 4 subunits, 2 alpha subunit, 2 beta subunit, which are held by disulfide bond. And I have already told you all, it is active, or insulin is active only when the the disulfide bond is intact. So let's go to the next slide, slide number 8. If you look at this picture, you can see very clearly. Let's see, I have told you all the insulin receptor has got 2 subunits. So you can see in the picture there is an alpha subunit which is present on the surface of the cell, and you have got the 2 beta subunits which is literally inserted into the cell, or you can put it as which penetrates the cell membrane. And the speciality of this beta subunit is it has got tyrosine kinase activity. Now, how is the relation here? So the beta subunit which has got, can you see in orange color the tyrosine kinase mentioned there. So the beta subunit which has got tyrosine kinase activity, the alpha subunit regulates it. So look at the third point on the PPT. The alpha subunit which is present on the surface of the cell is the regulatory subunit which inhibits the activity of tyrosine kinase. So when insulin hasn't arrived at this receptor, what is happening is the alpha subunit is inhibiting the activity of tyrosine kinase. When insulin comes and binds to the receptor, the inhibitory control of the alpha subunit on the beta subunit or tyrosine kinase activity disappears, and the cellular actions of insulin begins. Is that clear? Now the glucose enters the cell through GLUT2 and the process continues. So please keep that in mind, the alpha subunit. So here this slide is actually giving us a detailed description of the insulin receptor which is present on the cell membrane, and it is through this insulin receptor insulin is going to exert its control over the cell. Now the insulin receptor has got 2 subunits, the alpha subunit which is present on the surface of the cell, the beta subunit which has got tyrosine kinase activity which penetrates the cell membrane. Now the alpha subunit controls or inhibits the activity of tyrosine kinase until insulin comes and binds. Moment insulin comes and binds to, you can see the the dark green color molecule, the insulin molecule, the hormone which is come and bound to the alpha subunit. Moment that binding happens, the inhibitory control of alpha subunit on the tyrosine kinase activity disappears. Now we go to the next slide. So that's basically the actions of insulin. Now you look at the actions of insulin. First we discuss with the effect on carbohydrate. Now the effect of carbohydrate on insulin mechanism, we call it as hypoglycemic action. So insulin helps in the uptake of glucose in almost all the cells of the body, but more of glucose absorption is facilitated in the skeletal muscles, adipose tissue and liver. Please keep that in mind. Insulin secretion or insulin generally controls the glucose uptake into the cells or tissues in most part of your body, but you can see that a more pronounced influence on glucose absorption is seen in skeletal muscles, adipose tissue and liver. And I've particularly highlighted in the PPT, there's a point to be remembered there, which is often a Viva question for you all. Glucose uptake is not influenced by insulin in 3-4 places in your body: in the renal tubules, the red blood cells, the intestinal mucosa, and the brain, except the satiety center in the hypothalamus. That needs to be kept in mind. So, the satiety center in the hypothalamus, to glucose to enter, insulin is required. Rest, all part of the brain, intestinal mucosa, red blood cells, and the renal tubules, glucose can enter them without the influence of insulin. Now, look at the point B. Point B tells you all that, see, basically body wants glucose to be trapped inside the cell. So, insulin inhibits the activity of glucose phosphatase enzyme, which is responsible for the conversion of glucose 6-phosphate to glucose. So, that's what the point B tells you. So that glucose is trapped inside the cell, body wants glucose to be trapped inside the cell. For that, insulin, what it does is it inhibits the activity of glucose phosphatase enzyme, which is responsible for the conversion of glucose 6-phosphate to glucose. Now, so that's about the effect of, or the hypoglycemic action of insulin on carbohydrate metabolism. We move to the next slide. No, I think there are 3 more points to be done with carbohydrate metabolism. It promotes synthesis of glycogen by activating glycogen synthetase. It inhibits glycogenolysis. It inhibits gluconeogenesis in the liver. I don't think basically glycogenolysis, gluconeogenesis and all needs any further explanation for you all. These have been concepts which we have been repeatedly using in many systems. So I hope that you know what the process means. In biochemistry you all deal in very detail with those concepts. So we move to the next slide where it talks about the effect on fat. So basically the effect of insulin on fat is, it is lipogenesis. So by increasing glucose, so how to put it in a single sentence is, the effect on fat, the lipogenetic action of insulin on fat is, by increasing the glucose utilization by the cells, it spares fat. That's why insulin is called as a fat sparer. Is that clear? Insulin, it will increase the glucose utilization by your cells and tissues, and what happens is the fat in the body or in your diet is getting spared. So that's why we call it as a fat sparer. Now when you go to point number 2, insulin stimulates the activity of lipoprotein lipase. And now what does this lipoprotein lipase do? This is actually, lipoprotein lipase is needed to split triglycerides and it increases the free fatty acids in the plasma. So for the splitting of the triglycerides, lipoprotein lipase is required, and this lipoprotein lipase is stimulated by insulin. Now the free fatty acids which are now in the plasma, which is released by the splitting of the triglycerides, the free fatty acids are stored in the adipose tissue as triglycerides. Insulin inhibits the hormone sensitive lipase, and this particular enzyme is needed to split triglycerides into free fatty acids. And another very important function of insulin on fat metabolism is, it increases the uptake of ketone bodies, and these ketone bodies are formed in the liver, and the ketone bodies, it increases the uptake of ketone bodies in the muscle as energy. We go to the next slide. which is the effect on protein metabolism. On the protein metabolism, we call it as an anabolic effect on the protein metabolism. I think that should be slide number 11. Insulin is called now the effect on protein metabolism effect, we can put in one word, is the anabolic effect of insulin on protein metabolism. So the point starts with insulin stimulates the transport of amino acids into the cell. Especially you can see that the amino acid transport into the muscle cells is really fastened or stimulated by insulin. The mRNA translation is increased. Transcription of selected DNA sequence is also increased by insulin. Catabolism of protein is inhibited. Now, how does insulin inhibit the catabolism of protein? By decreasing the normal degradation of proteins by the cellular lysosome. So, insulin keeps a strict check on the degradation of protein by the cellular lysosome, so that catabolism of protein is inhibited. It depresses gluconeogenesis. The substrate usually used are amino acids, so no, we are conserved and stored as protein. So basically pancreatic action on the protein is anabolic. So, and we move to the next slide. So since insulin causes storage of carbohydrates, fats and proteins, we call insulin as hormone of energy storage or the hormone of abundance. So now when we have gone in detail to what insulin does to carbohydrate metabolism, protein metabolism and fat metabolism, the concluding statement we need to make is insulin is trying to store carbohydrates, fats and proteins, which gives insulin the name, the hormone of energy storage, or the most commonly used word is the hormone of abundance. So let's go to the next slide, where the regulation of insulin is discussed. The main stimulus, see what you all should understand is the main stimulus of insulin secretion is glucose itself, and it acts directly on the beta cells. The secretion of insulin in the body, we call it as a biphasic secretion. It happens in two phases. The first phase of insulin secretion is rapid, but it is short-lived. You cannot call it exactly short-lived. You just put it as a short-lived increase in secretion. That is the first phase. A rapid phase where the insulin levels are high only for a very short period of time. Now the second phase is a slowly developing prolonged increase in secretion. So that's basically the two phases in which insulin is secreted in our body. So now to go to the applied aspects, insulin deficiency produces a clinical stage called as the diabetes mellitus. An excess secretion of insulin can cause hypoglycemia. We need to go a little detail about diabetes mellitus, which is a part of the essay question if glucose regulation comes as a 10 marker for you all, or diabetes mellitus is very often asked as a 5 marker too. So diabetes mellitus, so when the question comes, I think you need to put a proper definition of the situation there, and the definition please try to exactly put it how I have put it on the slide. Diabetes mellitus is a syndrome of impaired carbohydrate, fat and protein metabolism caused either due to lack of insulin secretion or decreased sensitivity of the tissue to insulin. So what the definition tells us is diabetes mellitus, the syndrome is with an impaired or a defective carbohydrate, fat or protein metabolism. Now a defective carbohydrate, fat and protein metabolism can happen in the body due to two reasons. It can be either due to a lack of insulin secretion by the pancreas, or the problem would lie with a decreased sensitivity of the tissue to insulin. The pancreas is secreting insulin in sufficient amount very actively, but your body tissues sensitivity to insulin is reduced, or your cells are not responding, or their response is lesser than the required amount in the body, which results in diabetes mellitus. I repeat or read out the definition once again. Diabetes mellitus is a syndrome of impaired carbohydrate, fat and protein metabolism, which is caused either due to the lack of insulin secretion or a decreased sensitivity of the tissue to insulin. Now, the predisposing factor to diabetes mellitus due to lack of insulin, we put it in 3 points. The first factor is hereditary. The most typical form is hereditary idiopathic diabetes mellitus. Age, disease is common with increasing age. Obesity, people who are obese has a tendency, or shows a more tendency for diabetes mellitus, and there is a clear cut reason for that. The adipose tissue which are more in obese, those adipose tissue is basically more resistant to insulin than a normal individual. So the circulating free fatty acids in the blood increases the insulin resistance in the liver and the muscle. So I repeat it, the adipose tissue in the obese people are more resistant to insulin than normal individual. The circulating free fatty acids in the blood increases insulin resistance in the liver and the muscle. We move to the next slide, where the types of diabetes mellitus is discussed. You have two types of diabetes mellitus, the juvenile diabetes or the type 1 or the insulin dependent diabetes. The second one is the maturity onset or the type 2 or the non-insulin dependent diabetes mellitus. So just see the first one, the type 1 or the insulin dependent one. It occurs before 14 years of age. Family history is normally uncommon. Ketosis and acidosis develops with the condition. Insulin levels are low. Type 1 diabetes is insulin sensitive, and the treatment normally used is injection of insulin. Now, when you look at the type 2 or the non-insulin dependent or the maturity onset diabetes, the most common type is that, and it normally occurs after the age of 40 years. Family history is very strong with type 2 diabetes. Ketosis is usually absent, whereas have a look at the type 1 diabetes, both ketosis and acidosis is present. Now, when you look at the insulin levels were low in type 1 diabetes, whereas if you look at the type 2 diabetes, initially the insulin levels are normal or increased, but later the insulin levels drop. This type 2 diabetes is insulin resistant, whereas the type 1 was insulin sensitive. And the treatment for type 2 is diet restriction, oral hypoglycemics or insulin only during infection. Now, a little more inputs on the condition. In both types of diabetes mellitus, the metabolism of the main food is altered. So the basic effect of insulin lack or insulin resistance, see, it can be both reason, either due to insulin, the less levels of insulin in the blood, or an insulin resistance which is shown by the tissue. The effect, whatever condition, the effect is on the glucose metabolism. So this prevents efficient uptake and utilization of glucose by most of the cells in the body. So what happens is glucose concentration slowly builds up in the blood, but it is decreased inside the cell. So what happens to cell? See, the primary source of energy for the cell is glucose. So, but the glucose is abundant in the bloodstream. But because of the current situation, the glucose is not able to enter the cell, and now the cell has to use the stored forms of energy. So basically this condition we call as the starvation in the midst of plenty. So you have lots around you, but you're not able to use it. Now let's go to the clinical features. See, the clinical features I have, put it in 10 points, really briefed up. So please don't try to miss or skip on any of the points. So the cardinal signs and symptoms of diabetes mellitus are, the first one, let's put it as hyperglycemia. If for anyone, if any of these words are not clear, I have, put the explanation in brackets. So hyperglycemia, which is increased blood glucose. Number 2, glycosuria. The presence of glucose in the urine is when we use the term glycosuria. Point number 3 is polyuria, where there is an increased urination. And what is the reason of polyuria in diabetes mellitus? It is due to osmotic diuresis. We have already discussed osmotic diuresis in renal physiology. Now the fourth point is dehydration. And why is dehydration happening? Because due to increased loss of water in the urine. Point number 5, polydipsia, which is increased thirst, which again the stimulus is the dehydration. So polyphagia, which is increased appetite. Now, why is the person feeling more and more hungry? Please try to recollect the point I told you, that for the satiety, for the glucose to enter the tissue of the satiety center of the hypothalamus, insulin is required. So when there is lack of insulin secretion, or the cells of the satiety center showing a resistance to the insulin entry, what happens is the satiety center is not getting feedback on the already existing high levels of glucose in the body. So what it does is it increases, or it gets the person into a mode of polyphagia, where the person tends to overeat. So that is polyphagia there. Now point number 6, loss of weight, because protein catabolism is happening and lipolysis is happening for energy, because cells are not able to utilize glucose. So cell is relying on breaking down of proteins and fats, so the person loses weight. Ketonuria happens. Now number 9 is poor resistance to infection due to protein catabolism, because the body is relying on protein and fats as the primary source of energy now, because glucose is not available to the cell. So I think there we end with the class. So please go through the slides, with the audio input, which you are getting through my sound, and please try to make your possible lecture notes. I think a few extra points I have told you. I'll only be with the beginning 2-3 slides. Rest, I have really briefed up, summarized the points in your PPT. So once 2-3 times you go through the slides, listen to the audio. Please try making one mind map which fits into one page, so that your study is complete. Okay, bye guys, see you. Nice time studying.